Now the next thing that we need to understand is electron transport chain. You were introduced to this term in the previous lesson. Now we have to understand what it is. Now one thing might come in your mind, that where is that photochemical phase, because we are studying mechanism of photosynthesis, and I had told you in the beginning that we study it in two phases. Nowhere there is mention of that phase. So I must tell you that we are studying these terms in order to understand that those phases in a better way, so that we don't have problem. So in order to understand them in a better way, we need to get introduced to few terms and a few phenomena that we have not understood till now, so that understanding of the mechanism in proper would be better. So in that context, we have another term that is to be understood, and that is electron transport chain. First thing that you have to remember is that it was proposed by Hill in 1939. The same Hill who had earlier contributed by telling that there was a Hill's reaction we had talked about, and this Hill person is responsible for the main reactions in study of photochemical phase as well. So when we talk about electron transport chain, it was formulated by this person in this particular year, and later on further studies were done on it. In the electron transport chain, we study from where the electron gets originated and where it goes till the end. Okay, so basically we are going to track the passage of electron from the site from where it gets excited and where it is trapped in the last. So firstly, it would be excited from the photosystems, that you have to remember that the photosystems would trap, the photosynthetic unit would trap the the energy which is present in the photons, and the entire unit would take up that energy, and it would pass its energy to the photosystem respective one, whether it is P700 or it is P680. That means photosystem 1 or photosystem 2 respectively. So the energy trapped by the photosynthetic unit would be transferred to the photosystems, which are two, that is PS1 and PS2. If we have to say it otherwise, the photosystem where the reaction center has this wavelength for excitation, and here the reaction center has this wavelength for excitation. When these photosystems, they take up the energy from sunlight, they reduce themselves, and they become, not reduce themselves, they become strong oxidants. Okay, they become strong oxidants. How? Because they give up their electron. Now from here, this would be the site from where the electron transport chain's study material would begin. That means these photosystems, they just got rid of their electron by taking up the energy from the sunlight. Their electron, it got beyond its orbitals and it got excited, and that electron would move on to a reducing agent, particular reducing agent. Now let us see in individual case. Supposedly we are taking the example of photosystem 2. A wavelength of 680 nanometer along with the accessory energy that was taken up by the accessory pigment would generate enough energy to make the electron possible to get released out of its orbital, and that electron in the case of photosystem 2 would be taken up by pheophytin. Remember this term. Pheophytin, as soon as it takes up the electron, it makes the photosystem a strong oxidant, that means which is devoid of an electron, ready to take up the electron from wherever it is available. And this pheophytin, by taking up the electron which was excited, in turn becomes a strong reducing agent. That means it has an extra electron and it is ready to give its extra electron to anybody who comes over there, and by anybody I mean another agent which would take up that electron. Now this pheophytin passes its electron to another one, that is plastocyanin. Not plastocyanin, plastoquinone, sorry. it passes its electron to another electron acceptor, that is plastoquinone, which in turn gets reduced. Then this plastoquinone passes on its electron to cytochrome B, cytochrome 6 process, cytochrome complex B6, this is cytochrome C complex, and the cytochrome B, cytochrome C complex further passes its electrons to plastocyanin. Okay. Now, these particular agents, they are taking up the electrons. What about the photosystem, which became a strong oxidant? It takes up its electron deficiency is covered by taking up the electron which are generated by splitting of water. So basically, it's photosystem 60, that is PS2, becoming a strong oxidant enables the splitting of water into electrons, and those electrons are taken up by P60, and somehow its thirst for electron is quenched, and it does not remain the oxidant as it was earlier. So what you see over here is that the photosystem 2 had its electron excited, which was taken up by pheophytin. Pheophytin in turn gave its electron to plastoquinone. Plastoquinone passed on its electron to the cytochrome B6, cytochrome C complex, which further gave its electron to plastocyanin. Now this plastocyanin is a water soluble compound that gives its electrons to photosystem 1. Now this photosystem 1 too, in turn, by taking up the energy from the photons, has got rid of its electron. Now this photosystem 1's deficiency of electron, it too had become an oxidant in the same way as photosystem 2 had become. So this photosystem 1 takes up the electron from plastocyanin. Now this electron which was excited from photosystem 1 in turn goes to the agent X that we had talked about. Now this agent X is nothing but it is a chlorophyll molecule which acts as agent X. It takes up the electron excited by photosystem 1. This agent X passes on its electron to membrane bound sulfur compounds, that is FeS. Now this FeS passes it on to the respective reductase complex, which enables the reduction of NADP positive to NADH. Now this is the entire electron transport chain. This is what happens. In between, there comes another compound, that is ferrodoxin, which is responsible for enabling this reductase take-up of electron, so that there is reduction of NADP positive to NADPH. Now I am going to repeat it all. This is little bit complex, but why I have dedicated an entire lesson to it, so that when we study the mechanism and the photochemical phase, you're quite aware of these things, what they are, how they are working, and especially in phosphorylation. The topic of phosphorylation is going to deal with these terms. See, we had said that the electron would be excited by taking up the energy from photons. That electron would be excited from the photocenter. The photocenter had chlorophyll A molecules which were excited at different wavelengths. One of the chlorophyll molecules which got excited at the 680 nanometer wavelength was part of photosystem 2, and the other one was part of photosystem 1. Over here, I would like to tell you one thing, that the naming of these photosystems 1 and 2 has nothing to do with numbering. They have been named according to the time at which they were discovered. Photosystem 1 was discovered earlier and photosystem 2 was discovered later on. That is why they are named this way. Otherwise, they have no particular, this naming and numbering has no particular influence or role in the process. Now, what we have to deal with is that some electron is going to get excited at some stage, and it has to go from one particular agent to other agent, which would act as the reducing agent. the first photosystem molecule which which gets rid of the electron would act as an oxidant. Now then that oxidant has to be supplied with an electron. So we have the process of splitting of water, that is the water molecule gets broken down into protons, it gets broken down into nascent oxygen, it gets broken down into electrons, and those electrons would supply the need of the strong oxidant that would be formed by loss of electrons. Now, this is a heavy sentence. We will come to know what we are talking about. But for now, just see the passage of electron from where it goes to where. The photosystem 2 takes up the wavelength 680 nanometer. It's accessory, those antenna and core molecules, they also enable the electron excitation. The electron leaves the pigment molecule, the first photocenter molecule, reaction center molecule, and makes it oxidant. Keep that aside. What happens to that electron? That electron goes to pheophytin. Pheophytin takes up that electron. In the meantime, the photosystem which was devoid of electron takes up its electron from the water molecule itself. Okay, its thirst is quenched. What about the electron that was excited? This electron goes to pheophytin, makes it a strong reducing agent ready to get rid of its electron. Now, who takes up that electron next? It is taken up by plastoquinone. Plastoquinone further passes it into chlorophyll B6 and chlorophyll C complex. This complex further passes it on to plastocyanin. Now, this plastocyanin is a water-soluble compound that takes up the electron and passes its electron to photosystem 1, which again in turn, like photosystem 2, had become an oxidant because the light is falling on photosystem 1 as well. It also takes up the energy, the wavelengths, and it gets excited. The electrons are excited from photosystem 1 as well. So, the photosystem 1, on becoming an oxidant, takes the electrons from plastocyanin. Plastocyanin had taken the electron from photosystem 2. How photosystem 2 quenched its electron deficiency? From splitting of water. Now this photosystem 1's electron, which was excited by photonic energy, where it goes? It goes to the agent X, which is a chlorophyll molecule. It passes on its electron to a membrane bound FeS compound. This FeS compound, with the help of ferredoxin, which takes up the electron, enables the reduction of NADP positive to NADPH. So, the electron that was excited from the beginning gets fixed up over here. And the last step is reduction. So, this is what is electron transport chain all about. I would advise you to watch this lesson all over again so that you get a little bit of idea what we are going to deal with in the photochemical phase.